Hi everyone. Today we are going to learn a very short lesson on the structure of DNA. So you're going to be following along with your notes, and I will give you some hints as when to write stuff down and when you should highlight something. So the first thing we need to know is, what is DNA? DNA is called a nucleic acid. So that is different. DNA is not a protein, and it's not a carb, and it's not a lipid. It is a different category called a nucleic acid. So it's totally separate from those other types of molecules. It's a molecule classified as a nucleic acid. This is DNA and RNA are really the only two types of nucleic acids. The monomer, so DNA is a polymer. So the monomer of DNA is called a nucleotide. Make sure you write that down. You should know that the monomer of DNA is a nucleotide. And the function is the genetic code for life. So what does that mean? That means that DNA is the instructions for all of our cells. So what exactly is a nucleotide? So remember, a nucleotide is the DNA monomer. So a nucleotide looks like the picture on the right. The nucleotide is made up of a phosphate. Here is the phosphate. It's usually a circle. And then it's made up of a pentagon, a sugar. This sugar is called deoxyribose. You need to know that. And then there is a nitrogen-containing base. That is this thing here, connected to the sugar, the deoxyribose. There are 4 different types of nitrogenous bases. So the base could be an adenine, which begins with an A, or the base could be a thymine, which begins with a T, or the base could be a guanine, which begins with a G, or the base could be a cytosine, which begins with a C. So there are 4 different varieties of nucleotides. You could have the A-type nucleotide for adenine, or the T-type, or the G-type, or the C-type. All right, so one thing you should be able to do is to draw a nucleotide. Do you remember what it looks like? Take a moment now, test yourself. Where are you able to draw what a nucleotide looks like? All right, what did you come up with? Your nucleotide should have looked something like that, a circular phosphate group connected to a pentagon, that is the deoxyribose sugar, which is then connected to one of the 4 nitrogenous bases. In this picture, it is an A, it could have been a T or a G or a C. Any of those would work. All right, so DNA overall, the polymer now, so that was the monomer that we went over before. This is the polymer. The polymer is a double helix shape, and there are covalent bonds linking the backbone. The backbone is made of sugars and phosphates. And then the bases are joined with hydrogen bonds. And then here is something very important to remember. When the bases are joined, the A is always paired with T, the G is always paired with C. And we're going to take a look at what that looks like in a moment. All right, so how well do you remember that very last slide? What parts of a nucleotide make up the backbone? And what parts of the nucleotide make up the rungs? Answer this question now. All right, the backbone is made up of phosphate and sugar, right? And those are joined with a covalent bond. The rungs are made up of the bases, and you should remember that there are 4 of them, A, T, G, and C. Here we see a picture of double-stranded DNA. Now let's analyze this picture closely. We've got 2 strands here. The first strand is right here, what I'm putting brackets around. This is the backbone. The backbone is made of phosphate and sugar, and phosphate and sugar, and phosphate and sugar, and phosphate and sugar, and so on, all the way down. What kind of bonds are going to join a phosphate and a sugar? You should know that that is a covalent bond connecting all the phosphates and sugar. Those are all covalent bonds. And then the rungs are made of bases. Here's a base T, there's a base C, there's a base T, and there's a C again. Notice on the other side of the DNA strand that it is basically all of the nucleotides are upside down. Whoops. Here is the backbone. There's my backbone again. And then there are my rungs. All right, so this is what DNA looks like. It's two strands. What kind of bond is connecting the two bases? There and there and there and there. Well, you should know that is a hydrogen bond. So two strands, the strands are connected via a hydrogen bond. The backbones are connected via covalent bonds. All right, here's another picture of DNA. This right here is an entire nucleotide. All right, a nucleotide is made up of a phosphate, which I've just highlighted, a sugar, which is right there, and then a base. There are hydrogen bonds. The hydrogen bonds are right there, connected to another base, connected to another sugar, connected to another phosphate. So notice the sugars and the phosphates are connected on a backbone. The bases are sticking out, and they're not really connected except to the sugar, and they're hydrogen bonded to the next base. So we have two strands running in opposite directions. Here's a nucleotide, there's a nucleotide. Here's a nucleotide, there's a nucleotide. So we go down the list. The shape of DNA was discovered by Watson and Crick, along with two other scientists named Maurice Wilkins and Rosalind Franklin. All right. So on the previous picture, label a covalent bond and a hydrogen bond. So you should label the covalent bond as part of the backbone and the hydrogen bond connecting the bases. And then another question, what is the complementary base pairing for the following DNA code? So this you may need to go back a few slides to remember. What you should remember is that A's are always paired with T's, and therefore T's are always paired with A's. G's are always, C's with always paired with G's, G's with C's. So that is the complementary base strand. All right, if you have any questions, let me know.